So when you have a bacterial infection, antibacterial medication that the doctors use to treat you often exploits the innate differences between bacterial and your own normal, healthy human cells. But in the case of cancer, the differences are so much more minute. They're very similar to normal, healthy cells. And so this challenge of treating cancer cells without also killing off normal, healthy cells becomes very difficult. Now, despite this, my goal is to understand the mechanisms that rewire normal, healthy cells and turn them into cancer cells with the hope and the goal that in the future we can use this information to come up with new cancer treatments. So I study a family of transcription factors. And transcription factors are proteins that bind to DNA and cause changes in gene expression. And this may sound like a scientific mouthful for all of you, but let's go back to family real quick. By family, I mean I'm talking about more than one protein here, and that much like you and your own family, there are ways in which these proteins are very similar to one another, but also ways in which these proteins can be very, very different. And that's why they were of interest in prostate cancer. They seem to have almost completely different effects in the case of prostate cancer. Whereas one acted much more like a red light, preventing cancer formation, another related protein did the exact opposite and acted like a green light, accelerating cancer formation. And so we wanted to know, why would these similar related proteins have such different effects? One experiment we did was to take like a high-resolution picture of what these proteins look like. Now, as I said, they're proteins that bind to DNA. So on my slide, you can see these ribbony proteins that are bound to DNA. And I've colored them red and green, so you see which are my red lights, my stop lights, and my green lights. But other than that, I hope you can appreciate how similar they look like to one another. So this helped us understand, okay, yes, they're related, but it didn't really give us any insight into how they might be different. Now, my next experiment will make sense to any good parent. We asked, well, who are these proteins hanging out with? Who are their friends? And this is where we found a key difference. In the case of our stoplight was very good at keeping away unsavory characters, such as this blue protein up here. Whereas our green light buddied up and they went about their mischievous behavior. In this case, turning on genes that are important in cell migration and metastasis, or the dissemination of the local tumor to distant sites. So I hope I've shown you a key example in which very minute changes between different proteins can cause a very different effect from preventing or causing cancer. And our next goal is hoping to understand at a higher level how this cooperation occurs so we can prevent it as perhaps a new treatment for preventing prostate cancer. Thank you.